begin by thanking the organizers of this Young Investigators meeting. It's my first meeting, and it's a privilege to be invited here to share one's experiences under the garb of a mentor. What's it with that? Probably it's a way of probably sharing some of the angst as well as the little happiness that one encounters over a certain period of time, which probably the others haven't traversed through, and then in that sense share some of these things, share some of these experiences, which I'll do. So I'm Ramray. I'm an associate faculty in the Department of Developmental Biology and Genetics, formerly known as MRDG at the Indian Institute of Science. I'm also affiliated with the Department of Bioengineering at the Indian Institute of Science. So I think I have 25 minutes, and so there will be a buzzer at 25. Can I also request maybe a show of hand at 22, 21-ish? Because I need to maybe spend a couple of minutes on my last lines just to sort of give my take-home issues. All right, so I've titled my talk Seeking Form and Patterns in Cancer Trials Towards Thrills. I left the triumphs for a co-mentor, Abhijit, who will be talking about that. I've still not reached triumphs. I'm still stuck here. So how many of you have read Sherlock Holmes in an unabridged format? Anybody? Quite a few. Okay, so those who haven't would know that when Sherlock Holmes has an opportunity to critique what Watson has sort of narrated, his case-solving stories, and he does so by casting aspersions on what Watson does, and says that if I had written those things, I would have done so in a very austere, stripped-down fashion, where I would just talk about the facts and how each clue and evidence leads to the next thing. So this is like writing the papers. This is like giving a technical talk. Whereas what Watson did was adding what we Indians would call adding tadka, or adding phodo, or adding what we call in Konkani as bhannu. And so today's talk would add the tadkas to a technical talk, which I would give in some other forum generally. So just before I delve into the science and the associated the penumbra of my science, a brief sort of chronological trajectory of where I went. I started my career with an undergraduate in medicine at the University of Calcutta Medical College, where I did mostly human biology, if I call it that. And then I quickly grew weary of human biology and wanted to go and do something as unmedical, as unbiomedical as possible. And so I went on to New York Medical College, but where I did not do anything medical. I started working on a classical developmental biology problem, which is how limbs develop using chicken limbs as a model system. And so I delved into a little bit of evolutionary developmental biology and developmental biophysics. Before then, again, the bug sort of started work. The bug got back. Sorry. Yeah, maybe I'll try that. Yeah. Okay. Good. Pointing into oneself. Thank you. Better than I. So then got back into a little bit biomedical stuff by getting back into looking at pattern formation, development, but in a postnatal context, looking at mammary gland development, and then sort of moving that mammary gland development and the principles of mammary gland development into understanding breast cancer. And then, of course, have been juggling a lot of things, including science, at the Indian Institute of Science as an assistant juggler and an associate juggler at the moment, and doing, again, mostly human biology in all its strange dimensions. So, so what do we work on as a group? We basically what I've done as a group to sort of bring together a unique conceptual vision with which to sort of start a program is that I've tried to combine ideas, vastly divergent conceptual ideas from my graduate period and my postdoctoral period. And then sort of forged together a unique set of questions that one can ask as a part of my group at IISc. So we basically try to think about how organs, tissues, cells come together and organize themselves into complex multicellular entities as part of development. And then we typically think of cancer these days from a more microenvironmental point of view as the breakdown of this multicellular architecture, as a breakdown of tissues, as a breakdown of organs, such that cells can come out of that organ and go and spread to other organs. And so we sort of combine these ideas to then ask a question that puts things right on top of it by asking, does cancer then represent new rules, new patterns, new grammar to how cells can again come together and form interesting multicellular entities on their way towards other organs? Because now, with where cell biology has come through, we talk about organoids, we talk about spheroids, we talk about tumoroids, we talk about clusters, we talk about all kinds of interesting multicellular assemblages which come together in cancer in different parts of the body, maybe in circulation, maybe at the colonizing site, and so on, and seem to be responsible for causing cancer. So clearly, there is a grammar to cellular cooperation, to multicellularization, even in cancer. There is architecture in cancer. Is that architecture similar to what is seen in the normal form? Or is it distinct with its own rogue principles? Is this rogue morphogenesis as opposed to a more benign, divine form of morphogenesis, which we otherwise associated with development? So that's the broad philosophical vision of the group. And to break it down, we look at it from the point of view of different sort of theatrical locales as the cancer cell starts from its site of origin and moves towards the organ it's going to ultimately metastasize. So we try to recreate these theatrical locales by combining the matrix microenvironment within which these cells typically find themselves and play that video, if you will, under a microscope to see how cancer cells play with their matrix microenvironments, remodel them in a bid to come out. We could do that in an animal, but for some of the cancers that we study, such as ovarian cancer, it's difficult to witness this process in real time and in spatiotemporal dimensions. And that requires us to therefore recreate some of these processes outside, within certain approximating setups, and then try to study this process, study the kinetics of this process, study the dynamics of this process. So we can break, therefore, metastasis into a set of principles, such as the early migration from primary focus, the entry of these cancer cells from the primary focus into circulatory environments like blood, like lymph, like peritoneal fluid, and so on, the migration of these cells within these environments to newer areas, to newer organs, and ultimately the colonization of these cancer cells, where they decide, this is the organ I'm going to land in and thrive on. That's the organ that I'm going to leave and not thrive in. What decides these decisions, and so on. So, now I'm testing the distance. All right. So the time when I started asking these questions, framing these questions, was a very interesting period. Around 2015, when I started my group, was an interesting period in cancer biology, because at that time there were a series of seminal papers that had been written about how you had these things called circulating tumor cell clusters. Still pretty important to the cancer biology field because we've still not solved the problem. How circulating tumor cell clusters of cancer cells, move from the organ of origin and go and colonize newer sites. In fact, they seem to be having a greater propensity of being able to do that over and above single cancer cells. So everybody was on to this problem, everybody was excited, and I was no exception. And so the challenges that this gave me was, one of the things which I can point out as the fourth point was getting carried away. because my skills and my experiences were not geared towards being able to solve the problem at that time. But I got carried away. And I tried to follow this problem and quickly realized this cannot be solved by a classical cell biologist / developmental biologist. It requires engineering principles. There were big interdisciplinary groups in 2013 and 14, including bioengineers, physicists, and biologists working on this together. But who would tell that to me, who was crazily excited by this idea and wanted to do it? Spent time, lost time, dealing with this problem, and then quickly decided to give up. So I decided, no, I will not solve this problem for the moment. After about spending a year, my first year in IISE, decided to leave this problem for the moment. And then I decided, let me try to get back to a simpler setup. Let me try to ask the first question, which is, how do cancer cells come out of their primary environment, rather than how do they travel as multicellular entities to other areas? Other challenges, there are challenges such as grants. I wrote grants at that time. I think we are far further from where we were in 2015, where one of the grants that I've written and submitted to an online portal gets lost in the online portal. I can see it on my side. The grant manager could not see it on her or his side. Anyway, we are far from that. Chairs. I was fresh into the system and I applied for filling these positions, and I get CVs that have sets of principles, techniques, mastery over techniques which I had not even heard of. Okay? Again, solutions. What I realized, again, by making mistakes and assuming that everything that's on a CV has to run true, was that to next time be more cautious and be more careful in not trusting a person necessarily through the CV, but actually discussing and having a lengthy dialogue, discovering the strengths of what a certain individual can bring to a project, and then take it forward. Or in some cases, not select a particular person because there's not a good fit with respect to the kind of learnings that the student can bring to a group versus. And the third interesting thing, the challenge, was clinical collaborations. Yes, there are lots of clinicians in India, lots of hospitals in India. Everybody wants to collaborate. A lot of times the clinicians don't want to listen. And I don't blame them. I myself was a clinician at one point of time. The realities of being able to be privileged enough to practice medicine in India also means that the patient load one has to see is immense. And so an intellectual engagement with clinicians and with our clinical collaborators is extremely difficult. And one has to be very fortunate to get a clinician who will listen. Not just who will listen, who can listen, and who will listen. There are two different dimensions to that. And yes, there are lots of samples within these hospitals, but I quickly realized most of the samples that I was wanted to work with had been eaten by rats. And that's the reality. That's the reality. Government hospitals, which have these huge masses of samples, do not have the wherewithal to fund proper biobanking. It's the reality of being in the setup, and therefore one has to make peace. The solution is one has to make peace with the fact that one can collaborate with a private institution where you may not get the same numbers as you would get. You will not get patient samples to the order of 3 orders of magnitude. You will get far fewer patients, but perhaps the archival would be better. The banking would be better. The clinician would be less busy, and therefore with her or his clinical duties, and therefore would have time to listen to me. And so this is the trade-off. You can collaborate with a private institution, get less samples, and get more intellectual engagement with clinicians that I realized earlier by first jumping into a collaboration with a government setup and then sort of worming my way back into a private setup. Opportunities, accidental conversations. So going to the hospitals, chance conversations with other clinicians who are just sitting in ethical board meetings, immensely helpful. And so it's extremely useful if one were to be doing translational research, to be actually getting to a hospital, looking at the patients, talking to the clinicians, and then sort of getting both an intangible as well as tangible benefits from being inspired by being within that milieu. And of course, interdisciplinary discussions were crucial towards being able to try and ask some of the questions that we did towards understanding that first question of how cancer cells migrate early on from the primary focus. Again, bringing together a mixed group of students, including graduate students, JRFs, undergraduate students, master students, helped immensely in being able to develop some of the assays that we did. And again, coming to this point, it was crucial. It was crucial for somebody starting out like me to be able to therefore develop certain unique tools, certain unique assays that one can own and call one's own, and be able to therefore run a program with these assays. So these allowed us to sort of move beyond what I was working on in my postdoctoral stints towards making simpler three-dimensional assays, towards making far more complex hybrid three-dimensional assays, combining extracellular matrices, combining cancer cells, building more and more histologically mimicking setups. And those were the assays that have stood the test of time. I also at a very early time was also able to sort of use some of my understandings of computational cell biophysics towards developing certain multi-scale computer models, which helped me keep constantly looking between experiments and theory and be able to therefore make certain predictions with theoretical simulations which I could bring back into experiments. And this sort of dialectic between theory and experiments has richly served me, at the least, in being able to understand how cancer cells interact in very rich ways with their surroundings, in being able to come out in a three-dimensional context and then therefore get into the circulation. Coming back to the original problem of the multicellular clusters, which I had to leave, but it had its own time, and that time soon came when I was reasonably well known, at least within my milieu, within Indian Institute of Science, to be able to now talk with my engineer colleagues, with my physicist colleagues, and we then decided that, okay, now is the time where we can attack the problem of circulating tumor cell clusters and try to understand where these clusters come from. By that time, of course, circulating tumor cell clusters in blood was solved. So when we went to an ethical committee meeting in Kidwai Institute, that's the biggest government setup in Bangalore, somebody said, why don't you work with ovarian cancer? I had not at any point of time harbored any desire to work with ovarian cancer, had not even thought about it. But that chance conversation set up something very interesting, set a very interesting train of thought in my mind. Ovarian cancer doesn't spread through blood predominantly. It spreads through the peritoneal cavity, which is a cavity that's present in the center of abdomen. Okay? And so this cavity, unlike blood, this cavity contains a lot of fluid. So while it's very difficult to ask a clinician to take more than 10 ml of blood from a cancer patient, ovarian cancer patients come with a bloated abdomen because the fluid gets filled up in the abdomen. And so I can take out 2 liters to 3 liters of circulating fluid, which carries within it circulating tumor cells, often clusters. And so these were the first set of cells that we looked under a microscope in our lab from a patient who had what is called malignant ascites, which is the fluid of ovarian cancer. And the moment we saw these cells under the microscope, we saw that they were clustered. So they were circulating tumor cell clusters. Finally, I was on to solving the problem that I wanted to start my career with. And not just that, it led me to discover something completely new, which I have to thank my developmental biology background for, which is that many of these clusters were hollow clusters. And hollow clusters to a morphogeneticist or an anatomist or a developmental biologist worth her salt is basically indicative of organization. rogue morphogenesis. So now these cancer cells, which should actually destroy architecture, destroy organ formation, are almost coming to be organ-like within the abdomen of the patient. These are not organoids that one makes in the laboratories. These are de novo clustered organoids present within the body of the woman who carries it. So there's organization that cancer mounts in order to survive its traversal within a harsh fluid environment. And that, of course, led to the second, is that the 21-minute mark? I hope so. Okay, great, thank you. So I'll start hurrying. And so that led to the second program, where we basically were able to identify a new set of phenotypic heterogeneity that exists within ovarian cancer patients and try and understand how ovarian cancer uses multicellular clustering, rogue morphogenesis in order to survive within the abdomen of the patient and ultimately colonize the tracts. And so this has led to, over the last 6-7 years, using a interdisciplinary set of approaches which combines three-dimensional organ tech setups, integration with microfluidics, active matter physics, all in collaboration with colleagues towards trying to understand difficult questions which cell biologists, developmental biologists, at least of my limited experience, would not be able to solve by himself. Okay, and that's been useful. Now, coming to the point of international grants, I just wanted to talk about one story. Maybe given the time being slightly limited, I'll rush through one story, which was again helpful in my case. I generally write slowly and I write less, but I happen to write a commissioned review on the heterogeneity of extracellular matrices. Didn't seem to be received so well by generally the crowd that I thought would read it or would want to read it. But all of a sudden, after the review was out, I was contacted by two scientists at Marseille, and they were extremely excited about it and said, why not let's write a CEFIPRA grant together on this. In 10 days, based on that review, I wrote a grant with two people who I had never known before in my life. We got together, we wrote the grant. When the grant was over, Claire, who was one of the individuals, said, okay, why don't we write an international emerging action squad? I'd not even heard about the grant. It's a grant that CNRS gives its individuals towards being able to sort of get some soft money towards taking the leap towards bigger grants. Fine, no issues, we'll write the second grant. So we wrote the international emerging actions. In fact, we wrote a third grant, which is supposed to be an intermediate between CEFIPRA and international emerging actions. And lo and behold, we actually got all three grants. So a chance review not aimed at the community that actually noticed it or individuals that actually noticed it helped me get an international grant. So maybe luck was on my side as far as this is concerned, but maybe the take-home message for myself is keep writing, keep communicating thoughts, if not data, and it always can serve one well, perhaps in the days to come. This is another story, but I'll keep things optimistic here. The EMBO story is actually a good one, but I think Leo would be here, and there would be a talk on the EMBO Global Investigator Network, which I'm part of. So I will leave this one as well, except for saying please do apply for this grant. It's a tremendous grant for being able to forge collaborations and networking in the future for Indians. All right. Responsibilities of science. I'll end in 2 minutes now. Responsibilities of scientists. Teaching, administrative, outreach, and all three have served me immensely going forward. Teaching, of course, because it's allowed me to come in touch with extremely talented undergraduate students. Administrative, because I have understood it's important to be the change if one wants to make the change. And also, more importantly, to quote Bob Dylan, if one is not able to make the change, better stay away from it or move aside so that somebody else can come in and make the change. So don't stick to things that you can hold on to and you want to hold on to even though you can't do it. And of course, outreach, which is immensely important in terms of giving a satisfaction that I don't think papers and grants give. And lastly, the takeaways. So the takeaways are research well, flexibility has been important for me. These are takeaways for myself. an advantage of being able to be a mentor here is basically I get to sort of give myself a feedback of what I've been doing for the last 8 years without thinking about it. So flexible programs have helped me immensely. Grants preparation has helped me immensely. I have given my grant, each of my grants, I have given them to senior colleagues of mine whom I felt were able to judge them, and that has tremendously helped me get those grants, the ones that I got, of course. Laboratory life, diversity, inclusion, and equity has tremendously helped make my lab life a culturally rich experience. Administrative, departmental and institutional life, developing a peer or mentor group if one does not have, I think Shashi mentioned that in the morning, is important. Through outreach to empower all around us. And finally, I think it's fun to have a little bit of quirkiness in lab. So we have 2 lobsters as our lab mascots. And they gave us, I think they seem to embody all the things that I've mentioned here, including the bigger one trying to empower the younger one from taking a leap and so on and so forth. But they also apparently are the future models for cancer biology, because they seem to be 100 years old. They can be 100 years old. They don't age and they don't get cancer. So something for the future for all of us to think about. And in this, thank all the agencies that have supported all the problems that I faced with have stood with me through these times and helped me make the discoveries that I could make along the way. I'll stop. Thank you.
Thank you. Some interesting stories right there.
I'll tell more over other non-official forums.
Maybe one-on-one or in small groups. All right, we have time for a couple of questions before there's tea and coffee and some snacks waiting outside for you. So, sure.
So I had a quick question on that microfluidics thing you showed.
Yes.
So I mean, that seems very interesting because you can sort of tell what should be the cluster size which can navigate through a certain opening. So have you been able to find some rules on like size versus
So yeah, we can talk much more about it, and I find it hard to make me stop talking about it. But basically, that's the idea is for the microfluidics was to develop a correlation between deformation and size. Certain structures, which are elastic structures, will deform, but they will also, that will happen in a size-dependent manner. For other structures which are poroelastic, you would basically get a non-correlation between deformation and size. So it allows us to make a rheological distinction between different materials. So that story of purportedly on cancer biology has a lot of material biology or as well as mechanobiology.
I was thinking this could also act like a sort of assay to look for new drugs which actually can act on
We are doing that.
viscoelastic.
We've set up drug screens on such assays as well. Thank you for it.
Very good.
Yeah, hi.
Yes.
So I'm not from cancer biology field, but I was interested to know whether it is always the fixed sequence of organs that are affected in metastasis, or it depends on...
It's a question of what is called organotropism, where you have basically different organs being selected for the colonization by different organs. But I think also in the point that you're making, metastasis was thought to be a very linear kind of a story where you move from one organ to the other. It's not the case. There seems to be some kind of a parallelism rather than a series circuitry when it comes to metastasis. I can talk much more about it.
And could there be factors in the blood that also decide?
Yes, they do.
Yeah.
Platelets help, and a lot of other things are.
Thank you.
Yeah.
Last question.
Thanks, Ramray. Very interesting talk.
Thank you.
So, apart from the science part, did you try to do something about the biobanking program, which you faced problem here, or you just left it like?
No, that's a brilliant point that you bring up. Unfortunately, as you know, experimental biologists sitting within the academic institutions from our side, it's not easy to do the biobanking at our end. So we can of course do the biobanking at our end by storing the cells, storing the RNA, storing the DNA, and so on. But I think when I was starting out, the issue was more about the fact that if you get an ethical permission from a patient or taking her tissues for a particular research question, you cannot use it for something else. But now, with evolving conversations, clinicians are able to take ethical permissions for biobanking the samples. So now things are far better than when I started out. So now, in fact, a lot of hospitals have structurally incorporated biobanking protocols and practices within their setups. So you should, if you are interested, you should get in touch with those hospitals, and now you can use some sample retrospective, which is stored retrospectively for another project towards a different project as well. So that exists.